with the first talk by Dr. Elena Camacho Aguilar. So Elena is a native of Seville, and she did her bachelor in maths from University of Seville, and then went on to do a PhD at University of Warwick on interdisciplinary mathematics, where she worked on stem cell differentiation, doing dynamical systems modeling. I guess pretty similar. This talk would be pretty similar to the previous one. And she followed this up with a postdoc at Rice University, where she joined in 2018, where she essentially worked on developing experimental protocols, data analysis tools, and mathematical models to understand early human development. And starting in 2023, she has become a group leader in the Andalusian Center for Developmental Biology, which is back in Seville, and where she continues her interest in understanding differentiation of stem cells. So without further ado, let me invite Dr. Elena Camacho Aguilar to give a talk on building dynamical landscapes of cell-fate transitions. So Elena, on to you.
Thank you so much, Citabra, for the nice introduction. Good afternoon, everyone, and thank you, Shakti, too, for inviting me to speak at this wonderful conference. I wish I was there in person, but thanks so much for giving me the opportunity to present online. So today I'm going to be talking about mainly my PhD work in the group of David Rand, building dynamical systems models to understand cell-fate transitions. And I wish I could have joined the previous talk, but I think it's going to be similar. Yeah, but feel free to ask me any questions you may have. So what is cell differentiation? So cell differentiation is a very important process during the development of an organism in which undifferentiated cells, such as pluripotent cells, change their state to become more differentiated ones or specialized ones, such as nerve cells or muscle cells. And these transitions happen sequentially. As you see in this video, from a single cell that divides and divides, many different cell types arise, which make the structure of a more adult organism. And so the question is, how do these myriad... Oh, sorry.
Sorry, Elena, I think we are not seeing slide transitions. We are still seeing the first slide.
Oh, really?
Yeah, maybe you have to share your entire screen to be able to see, for us to be able to see this.
Let's see, share. Oh, we can, sorry. Yeah, so as I was saying is, yeah, how do these variety of cells arise, and what does it mean that a cell differentiates into different types, right? So, well, as I said, the cells that are in our body all came from a single cell. And so all inherited the same information that is encoded in the DNA. That the DNA is a very long sequence that divides into chunks that are called genes that encode for a particular protein. And these proteins are the ones that allow cells to perform functions. So even though all of the cells inherit the same DNA, not all of the cells produce all of the proteins. And as I was saying, the proteins that they express allow them to perform different functions. And so as the fact that a cell differentiated to another cell type means that the cell changed their gene expression to produce other proteins that will allow them to perform other functions. So as I was saying, so let's say that a cell is undifferentiated, it expresses a gene A. So the fact that a cell differentiates and becomes other cell types means that they might start expressing or change their gene expression to express other genes. So in this case, the first one would start expressing gene B, or it might become another cell type green, which is green, and start expressing gene C and gene D, and so on. And so from a single cell, many different cell types will arise. And so these transitions are controlled by cues in the environment, such as signals, which direct cells to become one cell type or another. So if the blue cell receives signal X, it will become purple, and with signal Y will become green. So the most common approach to try and understand how this happens in embryos, so the most common mathematical approach is to use gene regulatory network models, which model sets of genes or molecules and interact among them and control the expression and therefore the differentiation of a cell. As an example of a very well-studied system in biology to model cell state transitions is the differentiation of the vulva in C. elegans worm, where in this process, partially differentiated cells called vulval precursor cells, the gray cell here, differentiate into one of these three cell types, which are recognized by the expression of different genes. And these transitions are controlled by two signals, EGF or Notch. And so the lab of Marianne Felix modeled this system of molecule interactions that could explain how the signals control the differentiation into these three cell types. And as you can see, although this model might give a lot of mechanistic information about how the molecules interact and control this differentiation, modeling this system requires a high number of differential equations that depend on many parameters. So in the end, understanding how the parameters control the different stability and how the system behaves when changing this parameter it's hard. So the question is, can we develop lower dimensional model that could give us the essence of how these signals control how these cells become one cell type or another? So an interesting metaphor that I'm sure you have heard about before is this one given by the renowned developmental biologist Conrad Waddington in the 50s, where he depicted a differentiated cell as a marble that rolled down a landscape of hills and valleys. And so this marble would be encountered branching points or decision points where the marble will decide whether to take one path or the other, therefore becoming one cell type or the other. And the final valley at which the marble would end up will define the final state of the cell. And the shape of this landscape is controlled by the genes that pull this lower dimensional space. They control the shape, therefore controlling the path towards which the marble moves towards. And so even though this metaphor may seem very qualitative, we can actually formalize it in mathematical terms. So the state of differentiation of a cell is represented by a position in a landscape, which will be given by a potential function, V, which will have as many minima as cell states the cell can have. So in this case, it's very simple. The cell will just stay undifferentiated or state 1, or it might become differentiated or state 2. The potential function depend defines a flow that controls where the trajectory of the cell is going to move towards. And so in this case, the cell will stay in the undifferentiated state because there is a mountain or a saddle in between the state, and the trajectory cannot leave that state. And also the potential function will depend on parameters, in this case omega, which will control the shape of this landscape. And so through omega, we will parameterize the signals that in turn will change the shape of this landscape, and therefore controlling where the ball is going to move to. So in this case, let's say the cell is not receiving any signal, so this parameter is going to be 0. And so this is the shape of the landscape. So as the cell is receiving signals, this parameter is going to change, changing the landscape until it reaches a critical point in which the undifferentiated state disappears, opening a new path for the trajectory to move towards the differentiated state, and therefore the cell will become differentiated. And so this critical state at which the undifferentiated state disappears is it's an important point where the bifurcation of the state happens. And so the key features of this model is the number of attractors and how the bifurcations are controlled by the parameters. One can think about more complex landscapes. For example, this is a two-dimensional landscape, while the first one was one-dimensional. And here the basin of attractions are colored accordingly. So the phase A basin of attraction is colored blue, and so on. And as we can see, the parameter omega controls the stability of the basin of the attractor A. And so as omega increases, the basin of attractor of A bifurcates away and opens a path for the cell to become either B or C. But one can think about more complex biological processes in which you may have more attractors or more cell states. So this formalism, this mathematical formalism, was pioneered by Eric Sija and Francis Gorson, where they applied it to the process of vulval development I talked about before, and the patterning of the Drosophila embryo. However, in their applications, they normally use very symmetric landscapes, which are hard to apply in other contexts where cell states are not equivalent. Also, the way they constructed it, them didn't really allow to understand how analytically how the critical points depended on the parameters. So we wanted to try, during my PhD, we tried to find formalism that allowed us to build landscapes to any biological system in a way that we could really understand how these parameters control the stability of the system. And this was the goal of my PhD in the group of David Grant. And surprisingly, we realized that the answer to this question laid in a theory that was developed by the mathematicians René Thom and Christopher Seaman in the 70s, that were actually inspired by Waddington ideas. And so just an important point of this theory is that it gives us this classification of main or basic catastrophes, where all the bifurcations that we can find in a dynamical system are going to be locally equivalent to one of these. And they are ordered by complexity. So the fold catastrophe is the simplest one, then the cusp, and so on. So we can think about using them to build more and more complex landscapes, as so we use them as Lego blocks to kind of build the landscape that we need to explain the biological data. And so as an example, let's look at the fold catastrophe. Another thing that I forgot to say here is that, as you can see, the catastrophes are polynomial equations. So if one wants to understand how the critical points depend on the parameters, it's easier, right? Because we can just find the zeros of these equations or the derivative. And so we can have a really analytical understanding of how the bifurcations happen. So as I was saying, as an example, let's look at the most basic catastrophe, which is the fold. And here, the fold is one-dimensional, but here to ease the later understanding of the model, I have drawn it as a two-dimensional landscape. So the fold happens on the y-axis. And so one can compute, as I was saying, the critical point depends on how the critical points depend on the parameter C, which is the catastrophe manifold M. And so we know that for any C, we will have this number of attractors or saddles. So and another thing that we can compute is the points of the parameters at which bifurcation happens, which is the singularity sets. And so we know that for C equals 0, the bifurcation, just at C equals 0, bifurcations happen. So with this study, we can know that for values of the parameter C, which are negative, we're going to have two critical points, one that is going to be stable, which is the circle and corresponds to this attractor, and another one that is going to be unstable, which is the triangle, and it's going to be here. I don't know if you see my cursor, but yeah, so for values of C, which are negative, we will have 2. And as we increase the value of the parameter C, these two critical points are going to get together until they bifurcate away for C positive. And so no critical points will be in the landscape. So having this understanding of the possible bifurcations that can appear in the landscape open us a new way of building Waddington-like landscape models for biological systems. So what we can think about is to characterize the cell types that we see in a biological process and how they may appear with this signals that are in the system. Then we can try and use catastrophe and dynamical system theory to enumerate the landscape that contain the desired number of attractors and the transitions. Then build these landscapes and write the control parameters of our functions of the biological signals, and then use parameter fitting to try and fit these models to quantitative data, and then validate them to see if they are actually reproducing the data and try and make predictions to further validate them and also find new conditions that may be interesting. All right, so to to try and explain better this methodology and how we used it for understanding different biological systems. The first problem that we tried to tackle was, again, vulval development in C. elegans, because it was a very well-studied process already. There was a lot of data available, and we already had proposed landscapes by Eric and Francis Corson, Eric Sijan and Francis Corson. So we wanted to try and see what we could develop. So as I mentioned before, during the process of vulval development, 5 vulval precursor cells, P4 to P8P, will differentiate into 3 different cell types, which are primary, secondary, or tertiary, which is non-vulva. And so these 5 cells differentiate in parallel, and this differentiation is coordinated by an external cell called anchor cell, which sends EGF signal to the vulval precursor cells, which in turn start expressing a secondary signal, a paracrine signal among them called notch. And so in the wild type case, which is the worm that we find in nature, P6P will become primary phase, then P5P and P7P will become secondary phase, and the ones that are farther will become phase 3 or non-vulva. And as you can see, the process is normally symmetric around the anchor cell. So in our study, we focus on just modeling three of them. And as I was saying, there is a lot of experimental data available. There is a set of data that are what kind of like the wild type outcome. So in the wild type case, as I was saying, P4P become tertiary, P5P become secondary, and P6P become red or primary. And you can do certain perturbations that don't change these outcomes. So for example, you can remove the EGF receptors in P5P, and that doesn't change what the P5P becomes, and so on. Then there are other perturbations that do actually change the wild type outcome. So for example, if we put 2 anchor cells in the system, P5P will become primary in this case. Or if we remove notch signaling, then P5P won't become secondary, but it will become tertiary. There are another perturbations which are called partially penetrant, where we don't get a deterministic outcome, but we will get different patterns depending on the experiment. So for example, if EGF is overexpressed, we might get different patterns, which proportions are shown here. And then another set of experiments, which I think are really important here, are the ones playing with time. So it gives us a temporal aspect of the process. For example, in this one, the anchor cell is ablated or removed at different times, and the pattern is looked at the end of the experiment. So it gives us information about how much time the system needs EGF in order to become wild type, and what happens if you remove EGF earlier. All right, so we know that if we have 3 cell types that the cells can become, there are 2 signals. So how can the landscapes be? So the simplest landscape is a one-dimensional landscape in which you have all the attractors in a line. So it's a sequential type of decision, which correspond. One can think about a morphogen model, right? Where you low signals, you become the default state, higher signal, you become another fade, and then high signal becomes the primary fade. It might have been mentioned before, but it can be considered as a binary choice landscape. And we knew that this one couldn't be because of the anchor cell ablation experiments. Then in terms of complexity, the next landscape would be one in which you have two decision points. So you either become non-vulva or tertiary, or you become vulva. And then once you decide that you are becoming vulva, you can decide whether you become primary or secondary. And this is similar to what now it's called the heteroclinic flip. And the third more complex landscape is one in which you can decide at any time between the three fates. So this is a symmetric one. All the decision points are allowed, and this is a more flexible, and it's the one that was proposed by Eric Sija and Francis Corson to explain the vulval data. So we came up with this other possibility, and we wanted to see whether this could actually fit the data and be consistent with the data we had. So to build this landscape, we wanted also to try to see whether using the very simple catastrophes as Lego blocks that we then paste together to find a more complex catastrophe landscape works. So we decided to use the fold catastrophe to explain the first decision process between non-vulva and vulva. And so the parameter C will control this decision, and then use the catastrophe to model the second decision, so between primary and secondary fate. And so this is the second most simple catastrophe. And the parameters are A and B. And depending on which value of the parameter you take, the landscape might have 3, 2, or 1 attractor. So if you are inside this cusp, you would have three critical points, which are 2 attractors and 1 saddle in between. And depending on the parameter B, one of the attractors would be more or less stable. So for B positive, you would have the red attractor more stable. And if you have B negative, you would have the green attractor more stable. And if you go out of the cusp, you will bifurcate one of the attractors. Okay, so then we had these two simple catastrophes, and we paste them with a Heaviside function to obtain the landscape that we were looking for. So on the y-axis, we had the fold catastrophe, and on the x-axis for y = 0, we had the cusp catastrophe. So this is like the heteroclinic flip landscape. All right, and the good thing of doing this with catastrophes is that we have a very analytical understanding of how the landscape looks like for any parameter value. So on the first row here, I'm showing you the bifurcation set. So the orange plane is the values of the parameter for which the fold bifurcation happens. And the the purple cusp cylinder is the values of the parameters for which there is a bifurcation on the cusp catastrophe. So we know for any values of A, B, and C, how the landscape or the flow is going to look like. And so now we have a mathematical model, but we need to link it to biology, so the biological process. And for that, we need to write the signals and as functions of we need to write the parameters as functions of the biological signals, which were AGF and notch. So we simply take a linear transformation between the signals and the parameters, and that defines a plane on the parameter space. And by intersecting this bifurcation, 3D bifurcation set with the plane that the signals define, we can actually get this fate map of which attractors are present for each value of the biological signals. Of course, these parameters of the linear transformations need to be fitted, and that's what we are going to feed to the data. And so we use, in summary, we use these two simple catastrophes to build up a more complex landscape that we could try and feed to the data. So we could choose some values for these parameters of the linear transformation rates, and we can find this fate map. And with this model, we can already simulate the system. We can add some random noise to the trajectories to try and reproduce these partially penetrant fates. And so in these simulations, the cells start from the default state, which is the tertiary fate that the cells become if they haven't received any signal. And so when we put the anchor cell there, the EGF signal that the cell that is closer to it receives, this signal directs the cell to become primary fate. And since it's becoming primary, it will signal notch to the nearby cell, which will become secondary, and the farther away one will stay tertiary. And so the model for a choice of parameters can reproduce, for example, the wild type pattern. Yeah. Right, so we might be able to find parameters that reproduce one experimental condition, but the thing is that we had a lot of experimental conditions, and we wanted to see whether this more constrained model was able to reproduce them all. And for that, we took advantage of approximate Bayesian computation, especially a variant of this sequential Monte Carlo, which was pioneered by the group of Mike Stumpf. And so in this fitting algorithm, we explored all the parameter space sequentially, which allows us also to parallelize it. If you want more details of it, feel free to ask me or go to our publication or the publications by Max Stumpf. But what it does is we give the algorithm some prior distribution for the parameters, which we can know because, or like have a hint of because of we can we know how the parameters control the landscape. This understanding, this analytical understanding of the of how the parameters control the landscape. So we can have a clue of how we might have to choose the parameters, but we can have it very relaxed so that the algorithm explore very broadly. So we give the algorithm a threshold, which is how far away we allow our simulations to be from the actual data. And so we choose this first threshold very high, or like high, so that the algorithm explores the parameter set broadly, and we get particles or parameter sets that are so we have a broad distribution of distances between our simulations and the actual data. And so this is the distribution of distances that I don't know if you see my cursor, but at the end of these four boxes, that's the distribution that of distances that we accepted in this first step. So we accepted n particles, and then we decide, okay, we're gonna, from now on, we think that epsilon1, it's a good enough distance. We're gonna discard all of the particles that are above that distance, and we're going to redraw from those accepted particles, but also perturbing them. So we are re-exploring around the good enough particles. And so we do that over and over and over, decreasing the epsilon that we choose until we obtain a distribution or posterior distribution that we are happy with. So it's close enough to the experimental data. And so using this algorithm, we were able to fit these experimental conditions. So the ones corresponding to the wild type patterns, some that were not wild type but were deterministic, and also these partially penetrant conditions. And also some of the anchor cell ablation experiments. So in particular, the L2 letargos, so where the anchor cell is removed very early, and the DU divided, which is a very particular time point in which if you remove the anchor cell at that time, P6P becomes either first one, secondary, or tertiary in 1/3 probability. And so by fitting those, we could actually validate the rest of anchor cell ablation experimental data, and the model reproduced those as well. But also using our model now that was fitted to the experimental data that I mentioned before, we could make new predictions. So, for example, with this model, we could reproduce some epistatic, propose some epistatic experiments. So, for half wild type EGF, the pattern was unchanged. And also for half notch, the pattern was unchanged. But if you perturb both signals to half the wild type, you would lose the wild type pattern, and you would get tertiary and secondary fades on the P5P cell. And this was nicely understood by looking at where the signal would be positioned on the bifurcation set. And so we see that both models, the previously proposed by Eric and Francis, and our more simpler model, both reproduce the data. So the question is there any difference between them? Could we find an experimental condition that might distinguish between them? So we came up with this experimental condition where if one puts EGF for a certain time, our model will predict that cells are in a position where they can either become primary or secondary. And from that position, if you add notch and wait for another time, we would expect with our model that the cells will become secondary. Using the same protocol, but simulated with a more flexible model, it would predict that cells will become primary cells, cell type, but then become tertiary. So we don't have worms, so we cannot do this experiment, but it would be nice to try at some point and see what happens. And so I don't know if you have any questions about this part of the talk. Um, but if not,
I just had a couple of technical questions about.
sure.
I mean, so the stuff that Eric and Francis did, if you wanted to do it using catastrophe theory, you'd use the elliptic umbilic, right? So
Mm-hmm.
is that something that you ever tried to do to fit the data rather than their model?
Mm-hmm. Yeah, yeah. I tried to use the elliptic umbilic. I have data about that because we wanted to focus on using two together. I my last simulations or like my fitting focus on our model. I have some preliminary data on that. I didn't get as good results, but I don't understand. I need to do more research or um, yeah, more fitting to try and fit the elliptic umbilic because it's yes, it is mathematically equivalent, but in the actual model by Francis and Eric, they restrain the attractors to be on a radius 1 from the center. And I believe that might be important for the fitting. While the elliptic umbilic, if you increase the parameters, your attractors move farther away. But I don't know if that's important or not. It's something that I am thinking about, but I don't have final results that I could discuss. I hope that.
I see. Okay, yeah, and the other part was about the notch. So I mean, a couple of things.
Mm-hmm.
One thing is, so in their thing, they had this right, they had this line in state space, which
Mm-hmm.
determined whether the notch signaling was active or not.
Mm-hmm.
You didn't need that at all. How the cell states are somehow linked to the notch signaling. And yeah,
Yeah, sorry.
yeah, no, so that was one
No.
part of it. Then the second was, if I remember, they also had autocrine signaling, right? And
Mm-hmm.
did you have to include autocrine signaling? Yeah, so those are the two parts of.
Yes, yeah, I use autocrine notch signaling, and I use this very similar notch activation as they use. Yes.
Okay, thanks.
Okay, so from there, there started to be more interest in landscape model. I thought I would highlight these two, although later on have been more publications. So my next part of the talk is going to be about another application of this methodology we built on a more complex data set, and also some very interesting publication by the group of Stumpf on actually trying to see how the choice of noise affects the landscape. Both in the vulva development and in the second application, we use a white noise, but it's interesting to think what kind of other noises, how these noises could affect the transitions. So yeah, on this publication, which was a really broad collaboration with the group of James, David, and Eric, we focused on understanding marine trunk development. So in this process, the epiblast cells will become either anterior neural or will become posterior, or caudal epiblast in this case. And caudal epiblast cells, or like sometimes are called neuromesoderma progenitors, can become posterior neural or posterior mesoderm. And this process of anterior posterior axis formation and neural and mesodermal differentiation is controlled by FGF and wind signals. And so our collaborators are the Francis Crick Institute. We're trying to understand this process in vitro. And the nice thing of doing it in vitro is that you can perform all of these signaling perturbations in time, and seeing single cells, how this perturbation might change how they differentiate as well in time. So we could look at how the signaling profiles affect how the cells are differentiating and what they become at the end as well. So Robert and James perform these experiments where they play with wind signaling through a chemical called cheer or chiron, and also FGF signal. And so in their protocol, they would put EGF from day 0 to day 3 to obtain epiblast cells, which then, if they don't receive any signal, they would become anterior neural, as marked by SOX1 and OTX2 expression. Here, there are a lot of markers. You don't need to remember all of them, so don't worry about it, but just to introduce them. So if you add chiron to the experimental protocol, that will induce posteriorizing fates. So you would get either neural or mesoderm, and it will depend. We saw that it might depend on the time that wind signaling is in the media. So what we had was a lot of gene expression data, facts, obtained by FACS or immunostaining. And so what one can do is from FACS or immunostaining to evaluate the expression of all these markers in single cells, and one would get a cloud of points in the expression space, which would give you information about what the cells are expressing and therefore what they are becoming. So we had all these perturbations of signaling, and we had data of what the cells were expressing at different time points. And so our goal was to understand how these signals were inducing the cell fates in time. Also, the differentiation of these cells. So first of all, we tried to convert this gene expression data in actual fates. The pipeline to do this was to use some of the conditions as reference data with which we will feed a Gaussian mixture model, which will serve as a classifier to then classify the rest of the conditions. And so with this, we saw that in order to get epiblast, SOX2 needed to be high, anterior neural express SOX2 high, and some SOX1. And so we classified the cells from their expression to these more discrete cell types. And so we saw in each day what kind of cell types we would see. And so we observed that there were two types of decisions in this data. So from the original epiblast state, cells could either become anterior neural or caudal epiblast. And we thought that we could maybe reproduce these decisions using this landscape called binary choice fate, where the cells can either become anterior neural or caudal epiblast. And the feature of this landscape is that you wouldn't see mixture of the two fates. So you either become one or the other. And the second decision process would be the decision after you posteriorize, whether you become posterior or, once you posteriorize, whether you become mesoderm or neural. And because we saw on the data mixes, so if you observe here, you either see anterior neural or you see caudal epiblast. You don't see a condition where you have them both. While here, we saw that under the same condition, some cells became neurons, some cells became mesoderm. So, and we could actually tweak that, as I will show you, by changing the signals. So we use a binary flip to model that transition. So cells that are caudal epiblast then can transition to this other part of the landscape and then will become either posterior neural or mesoderm in different proportions. So this is kind of a summary of the sequence of decisions that cells need to go through. And it's controlled by Wnt and FGF. And so we use the binary flip to model this first decision between anterior and posterior, and then the binary flip to model the mesoderm versus neural, and we merge them as in the previous case with this Heaviside function. That's a purple line between the two landscapes. So you move from one landscape to the other on the X. And so, yeah, we're using these two catastrophes or landscapes as building blocks to build a 5 attractor landscape. And so, yeah, if you could imagine this in 2 dimensions, that would be the landscape. And so the next step was to see whether this proposed landscape could reproduce the data. So we fitted the experimental data that we had, and we saw that our simulations really matched the experiments. Yeah, even we could see that we would get anterior neural and posterior neural. And so our question was, one of our questions was, can we try and come up with new experimental conditions that our collaborators haven't tried that could give us interesting outcomes? So one outcome, for example, is that we saw that the cells didn't remain in the caudal epiblast state much. So they were leaking to others, to the mesoderm or the neural state. And so one question was, can we find an experimental condition that could make these caudal epiblast states more stable? So we thought that maybe pulsing shear could do it. But our simulations said it otherwise. However, the experiments predicted that that actually these pulsing experiments will make the caudal epiblast state more stable. So we went again and tried to refine our model and try to see whether with this refined model we could reproduce this stabilization of the caudal epiblast state, and so we did. And the same with another experiment where we had a mix of anterior and posterior neural phase. And so once we had our refined model, we also tried to see whether we could give quantitative predictions. And one question was to see whether we could reproduce the fact that once that cells are anterior neural, they cannot become caudal epiblast anymore. So we tried to do that by inducing chi with winds late in the experiment. So one cells have already committed to anterior neural, and in fact, our prediction that cells won't become posterior anymore was true. And here I had put a video that Mary did that it's very nice. So cells start in the epiblast phase. When you remove FGF, they become anterior neural, and even though you put chiron or wind in the system, they will become anterior and they wouldn't de-differentiate anymore to the epiblast stage. And so I think this has been quite quick, but if you have any questions, we can go through them. But before I finish, I want to thank all the collaborators and colleagues that have been with me through these years. So David Rand, Eric Sija, and Francis Corson for their advice in this work. To my postdoc mentor, Ari Wormflash, and the lab for their support and advice as well. And as Dr. Citabra mentioned before, I started my own lab in Seville, in the Andalusian Center for Developmental Biology, where we are trying to work on understanding cell-fate decisions using human pluripotent stem cells and mathematical methods. And we are collaborating closely with other groups, both in the CABD and in Madrid and other institutes. I wanted to mention in particular Fernando Casares, Luciano Marcon, and Juan Poyatos, with whom we are trying to establish this supergroup called Adaptable Living Systems, where we are really interested in trying to understand biological mechanisms behind developmental biology. And so, yeah, now thank to you for listening to me even after lunch, which is a hard time. And yeah, I'm happy to answer any questions you may have.
Thanks, Elena. Questions? So we were having this discussion, just in the earlier talk. So the speaker was, in fact, seated right here, Chishman from the National Center for Biological Sciences, and we were discussing, what could be the mechanisms by which, over evolutionary timescales, these developmental landscapes change over time. And we are wondering if there is, like for example, in neuronal networks we have this learning rules like Hebbian learning and so on. And there has been speculations that adaptation is like the evolutionary analog of learning. So I'm just wondering, do you have any speculations as to, how over evolutionary timescales this, transitions of the developmental landscape, appear?
Mm-hmm. Yeah, that's a great question. I think that's very tricky. Yeah, I mean, I'm not sure if I'm understanding the question correctly, but actually fitting the time scales, it's one of the most difficult things I have found to fit the landscape models. So how to parameterize this correctly. I don't know if that's what you're asking to fit all of the different time scales within the same landscape. That's a good point, but I don't have an answer for that. It's really interesting.
So, for example, if you change the environment for the
Mm-hmm.
organism, sometimes it will alter
Mm-hmm.
its developmental trajectory. And
Mm-hmm.
so Archishman showed multiple examples where by sustained change in the environment, you can actually make a particular shift in the developmental trajectory permanent, so that even if you remove the environment, it now
Mm-hmm.
continues to, in the later generations, it continues to switch to this alternative developmental pathway. So we were just, speculating as to what could be the meta dynamics which leads to, these kinds of switching from one developmental trajectory to another developmental trajectory. Presumably, it's some kind of sculpting of the landscape itself, but over evolutionary timescales rather than over, a single
Mm-hmm.
individual's lifetime.
Mm-hmm. Yeah, no, I don't have any answer for that. I wish I could have been to his talk. It was really relevant. But yeah, I'm sorry, but yeah, I don't know what to. Yeah.
So in the vulval, in the C. elegans vulva development case, do you have a similar kind of experimental setting where by changing the environment, you can actually, make, so, you showed that you can actually
Hmm.
alter the fate of particular cells. So have there been experiments where, you do this over several generations and see if this kind of sticks so that even
Mm-hmm.
don't have the normal even if you don't have that environmental stimuli, subsequent generations are still switching to
Mm-hmm.
the fate from the secondary
Mm-hmm.
fate to the primary
Mm.
rather than remaining secondary. Do you know of any
Yeah,
such
I'm
experiments?
not aware of data for different generations of the same coming from the same worm, but it's really interesting because, for example, in human embryonic stem cells, we would reuse, I mean, human pluripotent, but any stem cell, right? Um we grow the cells in media, and so we assume that the daughter cells are going to be exactly the same as the parent cells, right? So if you're saying that some modification on the parent cell remains for the future generations, even though they have not differentiated, is that what you're saying?
So it's a
Um,
bit like that. there could be multiple explanations for this, as Archishman was pointing out. Like, for example, there could be that particular alternative trajectory was always there, but it was, like
Even.
had a much higher potential so that it was never chosen.
Mm-hmm.
And so when you alter the environment, you are actually making it more likely. And somehow, this is causing,
Mm.
alterations in the developmental landscape over generations.
Mm-hmm. Mm-hmm.
So we don't
Yeah,
know.
wait. Yeah, that sounds super cool. Like, what, for example, do you have an example for that? What did you want?
Well, I don't know if I have an example. I mean, I guess we could ask, have you ever tried to look at the, because I know Marianne Felix has, right, tried to look at vulval development in other kinds of worms.
Mm-hmm.
Um, I don't know, have you ever looked, I mean, I don't know what the quality of the data there is, but
Mm-hmm.
I don't know if you're aware of anybody who's tried to look at how these landscapes change, do that kind of a comparison between species. Um,
I've yeah, I've heard. I think Francis was trying to look at that. Um, but I didn't continue studying vulval development after that. I know Marianne Felix had. I mean, from Francis, that she had different data on different mutants, but I sorry, on different worm lines. Um, but I don't know if she would have that kind of data of multiple generations. Yeah, I know. I think I may remember that Francis was looking at that, but I'm not sure what kind of data it was.
right.
Thanks. Harish?
Hey, I had a question. So when
Mhm.
you look at these gene regulatory networks, can you, just by looking at the topology, can you predict which components are play more of a role in storing the patterns, and which ones contribute more in transitions?
Mhm.
Is there like a neat segregation, or is it more mixed? Like, do they play more mixed role in?
You mean on the gene regulatory network model,
Yeah.
like the one that I was talking?
So I think you show these mutations where some would lead to transition and some would lock the differentiation and wouldn't allow the cell to differentiate any further, right?
Oh, you mean in the last one when I was talking that they would these that the cells will look into the anterior neural feed and won't be differentiate further.
Yeah. So are there like
Uh,
topological indicators which tell you what leads to encoding a pattern versus what leads to transitions? What components?
mm,
Um,
I want to really know whether I'm understanding your question. So you mean biologically or in the model?
Biologically. So
Or
for example,
biologically, yeah.
so there could be one particular gene which induces frustration in the system and that leads to
Mm-hmm.
something like that.
Yeah, so there is a publication where they look at the noise of the data when they are transitioning to new fates. And they say that they look at gene and cell to cell variation, and they can see that the genes that control the transition become more variable when they are transitioning. So that's kind of a signal. I think it's gene to gene variation with respect to cell variation becomes higher when they are transitioning to a new fate. So that might be an indicator of what it's controlling the transition.
So
But
there's
looking,
more. I'm sorry, continue.
no, sorry, that I don't know what might lock the cells to a certain fate, but I'm not aware of whether there are indicators for that.
Okay. No, no, I was wondering if incoherence in the signaling or the, measures like frustration in the topology could be an indication.
Mm,
Issue.
yeah, that's a yeah, that sounds. interesting, but yeah, I'm not aware
Okay.
of any study on that. Yeah, thank you.
Any other questions or comments? All right. So if there are no further questions, let's thank Elena once more.
Thank you. I mean, I would love that, honestly. Yeah. Thanks so much for
Yeah.
inviting me.
Thank you for
Yes.
being here. All right.
Thank you. Have a
Bye.
good day. Bye.
You too. Bye.